Okay, let's jump back in to where we were. We were talking about lipid metabolism. So we just described how we break down a molecule of glycerol and turn it into G3P. And now we can talk about the other bit, the other part of our fatty acid. So let's jump right back into our drawing. So let's get a marker out and talk about the process of beta oxidation. So what we're really talking about today is we're talking about, right, remember we have a triglyceride, like these three molecules of carbon that are all linked up together in this, that molecule that's a header on this. And then we have a fatty acid chain that's coming down from each of these. And those fatty acid chains are going to be variable in length. So this is like a triglyceride, so we have three of these fatty acid chains. So these three fatty acids. It's a fatty acid that we're going to be zooming in on how we're operating when we're doing things like beta oxidation. Right? These fatty acids are anywhere from a couple carbons to low 20 carbons, right? So kind of the standard one is kind of anywhere from the range of like 12-ish to like 24-ish, right? So just so you can have a range in what we're talking about in terms of these fatty acids. And so we are going to lipolyze this, right? So we're going to go undergoing lipolysis. Lipolysis is going to take place, right, where we end up cleaving all of these triglycerides apart into fatty acids and turn them into free fatty acids. The glycerol goes off and does its own thing. So the glycerol goes off and does the process that we'd already talked about. And then these fatty acids are then cleaved off after we've undergone lipolysis. And when you're looking at each of these fatty acids, these are going to be like a long chain of carbons. So like each of these little bins in this little thing is a chain of carbon. And then it ends with a carbon. And I don't particularly care that you're really zooming in too much on your chemistry in this, but just so that we're all kind of on the same page when we're talking about this, it's a chain with a carbon and then some oxygens at the end of it with a little bit of a charge. That's why this is an acid. It has an electrical charge to it. So this is our free fatty acid. For this one, it is a, what is it? Let me count the carbons. 8 carbon chain, right? So this one's going to be a little bit smaller than normal, but still, this is our free fatty acid that we are now ready to undergo beta oxidation. Remember, we took that molecule of glycerol and we turned it into one single molecule of G3P, and then that G3P then just runs through the second half of glycolysis, runs through Krebs cycle, just like we would have done normally. This is basically half of a molecule of glucose, not that much energy. Fats, like lipids, like what we're talking about, are incredibly energy dense, not because of the glycerol molecule that becomes basically half of a sugar. They are energy dense because of these free fatty acid tails. So what we're going to do is we're going to beta oxidize this. And the first thing that we're going to do is we're going to take this free fatty acid and we're going to add in a coenzyme A, the CoA that we were talking about previously, the thing that we make an acetyl coenzyme A as part of the transition step before we go into the Krebs cycle. We do that here as well. To do so, we actually have to put a little bit of energy into this. So we actually have to use some of our high power batteries. So we're going to take a molecule of ATP and we're going to turn it into a low power version. We're actually going to go so low power that it actually goes to AMP. We're using up a lot of energy to do this, but we're preparing this molecule, and in doing so, we're preparing it and getting it ready so that we have a lot of potential energy to start unleashing. So the thing that actually gets made from this, so when we combine this free fatty acid with a coenzyme A with a little bit of energy to zhuzh it along, is we end up getting a molecule called acyl-CoA, which is just going to be a long chain of carbons, and then a carbon attached to a molecule of oxygen and attached to a coenzyme A, right? So it looks an awful lot like a free fatty acid. It's just we've bumped off one of those oxygens and added a coenzyme A into it, right? So this is our molecule acyl, not acetyl, but acyl-CoA. Right, so now we are ready to start breaking this molecule down. we are ready to do the beta oxidation process. And what we end up doing is we actually do beta oxidation. So what we're doing is we're actually cleaving the second carbon from the third carbon. And this carbon's the one that you see right here labeled. That's actually not technically one of the carbons that we're counting. It's this carbon here, 1, and carbon 2. Another way that you can say those names is you could say that this is the alpha carbon and this is the beta carbon. Right, so we're actually cleaving the alpha from the beta as part of this, which is why it's called beta oxidation. So we cut these 2 carbons out, and then these 2 carbons separate off on their own. Right, we cleave 2 carbons off of this, and then this becomes a 2-carbon chain with a coenzyme A attached. And in doing so, we are releasing some of that chemical energy that's binding these 2 carbons together. We're obviously unleashing that energy and can capture a little bit of it in the form of these low power batteries that are not going to be like all that useful. Again, these are going to be our weird batteries where we're going to be charging up a molecule of FADH and we're going to be charging up a molecule of NADH from their low power forms. So this is like nice to have, but we're still not actually really getting ATP out of this. Oops, NAD. There you go. So we're powering them up to their higher powered forms. That's nice, but we're not really getting direct ATP out of this. But the thing that we do end up doing as part of this beta oxidation process is you actually have to add in one more coenzyme A because we're going to end up having a molecule that's using 2 of these, is we actually split this acyl-CoA that say this is a 8-carbon chain, that's an 8 acyl-CoA. Well, we're going to split 2 of those carbons off, and then at the end of it, we're going to get another molecule of acyl-CoA. It's just this molecule of acyl-CoA is going to be a little bit shorter than it was before. So we still have an acyl group on the end of these carbons, but now if this started out as 8, then this is going to go to 6-carbon acyl-CoA. This is still an acyl-CoA. We still call it an acyl group. Right? I'm not going to we don't need to get into the differences between a 2 and an 8-carbon chain, right? The only thing that matters is this tail's getting shorter and shorter and shorter. And we've broken 2 of those carbons off, so they go off and become their own thing. They become a molecule that you're already familiar with. They become a 2-carbon chain linked up with a coenzyme A, right? So that acetyl group that you're already familiar with. And this is an acetyl molecule with a CoA attached to it. So what is this called? This is acetyl-CoA. This is the product of the transition step. I'm going to shrink my... Get out of here, me. Right. We are producing the same thing that we are making at the end of the transition step, where we take a molecule of glucose, we break it down into glyceraldehyde 3-phosphate, turn it into pyruvate. Well, and then that pyruvate loses one of its carbons and becomes a 2-carbon acetyl-CoA. This then just goes into the Krebs cycle, just like any other acetyl-CoA, right? Just like an acetyl-CoA that we're producing from glucose breakdown, this acetyl-CoA, this 2-carbon chain, we add those 2 carbons of that acetyl group to the 4 carbons of oxaloacetate, and then they come together to form that 6-carbon citrate, or the citric acid. It goes around the Krebs cycle, we spin a little bit of energy off of it, things are all well and dandy. This is us getting more and more ATP from this. Well, that's what we do with the acetyl-CoA. How do we manage this other part of this, right? Now I've got a shorter chain, but I still haven't really gotten that much energy out of this. I've broken off one of these carbon groups, these acetyl groups. So let's just run it back. Just do it again, right? We started out with a 6-carbon group. We run that through. We add in a coenzyme A, and we can do this without adding more energy into it because we're actually already getting a little bit of energy out. We're powering up FAD, powering up NAD, producing a 2-carbon acetyl group, and then now the thing that we end up with at the end of this is now a 4-carbon acyl-CoA. Well, what do you want to do? You just run it back again. We run this right back through beta oxidation one more time, and the only difference being here at the end of it is we don't end up with an acyl group anymore. We end up with 2 molecules of acetyl-CoA, right? So every single pair of carbons that are in our fatty acids, right, however many we started with, if we're starting with a 16 carbon fatty acid, right, how many molecules of acetyl-CoA am I snapping off eventually, right? I'm eventually getting 8, right? These taking whatever this molecule is and dividing it by 2, right? So if I had a 20 carbon free acid, right, you can don't have to be a rocket scientist to figure out I'm going to get 10 molecules of acetyl-CoA out of this. I get a little bit of energy, right? It's nice to get these weird batteries, but the acetyl-CoA running around, getting some more of these weird batteries and getting a little bit more ATP. I'm going to take all of this NADH and FADHs that I'm producing and then pump them into oxidative phosphorylation, making even more ATP. This is why a triglyceride molecule is energy dense. It is not the glycerol molecule, it's the fatty acids. Well, hey, if I have a triglyceride that's a 20-chain fatty acid for a triglyceride, remember, I have 3 of those. It's a triglyceride. So I have 3 of these 20 molecules of carbon all in a row. So I'm going to end up getting 10 times 3, right? One for each of those fatty acids that's being cleaved off, right? That's where we're starting to actually really get a pretty substantial payoff, right? So as a little summary slide, right, so that everybody's all on the same page, we have our triglyceride. And this is going to be a process by which we kind of do a little summary and then add some more onto this. We have our triglyceride. That triglyceride is a molecule of glycerol attached to 3 molecules of fatty acid. Right? What do we do with that triglycerol or that triglyceride? We lipolysis. We cleave it apart, and then that glycerol goes off and becomes a molecule of G3P. That G3P goes off and becomes a molecule of pyruvate, just like we would do any other molecule of glyceraldehyde 3-phosphate. That pyruvate then goes through and eventually goes through the transition step and becomes a molecule of acetyl-CoA. Well, this is that glycerol breakdown that we just showed on Monday. This is the process of beta oxidation that we just described, where we take a fatty acid and break it into lots and lots and lots of these acetyl-CoAs. We could take this molecule pyruvate, like we had talked about previously. We can take pyruvates and then actually the liver can stitch them back together, right? So we talked about the Cori cycle. We could take this pyruvate and stitch it back together and produce a molecule of glucose out of this, right? This acetyl-CoA will then kick around that molecule of oxaloacetate, and turn it back into a molecule of citrate. And we run this around again and again and again. The bouncing ball that you have to follow is a molecule of glycerol is 3 carbons. Fatty acids are, I don't know, lots, right? So 20-ish, right? Molecule of glyceraldehyde 3-phosphate becomes a 3 molecule. That pyruvate is 3 molecules in a carbon, but then when we go through that transition step, it breaks it down to 2. That oxaloacetate is 4. 4 plus 2 equals 6 for our citrate molecule. If I want to undergo part of that Cori cycle and turn that pyruvate molecule, I actually have to put 2 of them together because that molecule of glucose is, of course, a 6-carbon chain. So this is how we're doing things when everything's going well for us, right? If I am starting to be activating a little bit of my lipid supplies, right, I don't have to be starving to have that happen. But there can be some other ways that this kind of gets messed up a little bit. If I, for whatever reason, stop consuming a lot of carbohydrates and I shift my energy intakes almost exclusively to things like fats, this is example, the keto diet. If you're familiar with keto diet, what this actually is doing is I'm basically not giving myself any molecules of glucose, and I have a hard time eventually running out of oxaloacetate. If I am glucose or carbon starved, what I'll actually do is I'm actually going to start consuming my own oxaloacetate, and I'm going to consume my oxaloacetate and convert some of this oxaloacetate back to glucose. I do this because I want to hold off a little bit more glucose and increase the glucose availability, especially for places like my brain, right? So I'm going to actually kind of recapture some of this oxaloacetate and through a process turn it back into glucose. That's not great for the Krebs cycle. If I don't have oxaloacetate, if the cells in my liver or skeletal muscle or whatever don't have oxaloacetate, I could have all the acetyl coenzyme A that I want, but I can't ever add those two back together to produce that 6-carbon citrate. So if I'm in ketosis, right, if I am basically starving myself of glucose, starving myself of carbohydrates, I am going to go through a process by I'm going to take these acetyl-CoA, which at this point, if I don't have the Krebs cycle, this acetyl-CoA becomes an endpoint, it's a dead end, right? I don't have anything to do with these acetyl-CoA. And what I'm going to actually do is I'm going to undergo a process called ketosis, right? So this is if things are this would happen if I'm starving or if I'm on some weird diet where I don't intake a lot of carbohydrates, say for a week or so, right? I'm going to start going into ketosis if I'm absolutely no carbohydrates being intook. Well, what that means is I'm going to take this a molecule of acetyl-CoA and I'm going to turn it into a molecule called a ketone or a keto acid, right? This is a group of molecules. I don't particularly care that we know all of the ketones, but the ones that we need to be a little bit familiar with is we need to be familiar with acetoacetone or acetoacetate. Acetoacetate is the main one that we turn these acetyl coenzyme A's into. We staple 2 molecules of acetyl-CoA back together and it becomes a 4-carbon chain. So we're back up to 4 carbons. Most of the time, we don't really have anything to do with this. We don't really use this in any way. We'll just urinate this out. This is not a big deal under most circumstances. Some of this acetoacetate is actually going to spontaneously, all on its own, break down to a molecule of acetone. So this is a 2-carbon chain, right? So it'll break down and become 2 molecules of acetone. But that's actually relatively useful because acetone is the thing that you take your nail polish off with, right? So acetone, if you start having higher levels of acetone building up in your blood because you're in ketosis, because of the diet deprivation of carbohydrates, you're actually going to start breathing out acetone in your breath. You're going to start blowing acetone off as your acetone that is dissolved in the blood switches to being a gaseous form and it comes back out from your alveoli. Right, so this acetone was going to start giving you kind of a fruity smell. It's not going to smell like nail polish remover because it mixes up with a bunch of other organics and stuff. But this is becoming a really, really big problem. If you are somebody that is in ketosis, you are normally, right, the solution to this is to pee them out, right? So normal individuals are just going to urinate these away. Right, you're going to regulate your ketone in your diet, so or in your blood, mostly by just excreting them. It's a waste product. We don't want it. It's an end point. I'm really just trying to harvest that coenzyme A and then send that coenzyme A back off to other systems that are using it. And then I'm going to throw those carbons away. That acetyl group is trash to me at this point if I don't have the Krebs cycle. Well, what happens if you don't urinate these out? What if you are somebody that has bad kidneys, right? So poor kidney function means that you're not able to actually excrete these out anymore. If you're somebody that has poor kidney function, you're going to have these acetoacetates and acetones and beta-hydroxybutynates, these other ketones building up in your blood, right? That becomes a big Problem. Now we have an issue. You start developing ketoacidosis. Oops. Right? When you are, these ketones are also called keto acids. So if I start having high concentrations of these keto acids in my blood, I'm dropping my blood pH. And as I drop my blood pH, I now could potentially be pushing myself out of homeostatic equilibrium. I could actually put myself into a coma, right? This is something that happens quite often when you are somebody that has diabetes. Because what do patients with diabetes have? Well, they have poor kidney function. Check. They generally have pretty bad kidneys. If it's unregulated and unmanaged diabetes, because diabetes destroys the blood vessels of the kidneys themselves. So then what are you going to get? Well, you're going to get ketoacidosis, but we give it a special name when it's a diabetic that has it. It's no real difference, the side effects, but it's just diabetic ketoacidosis. This is a pretty dreaded disease called DKA, diabetic ketoacidosis. Right? That's why we have to be really careful about patients that have poor kidney function if they have altered diet. You should not go on a keto diet if you're somebody that has poor kidney function, because you're going to put yourself in ketoacidosis, diabetic or not, right? So this is a part summary, but then if you are in somebody that's undergoing ketosis, then you'll actually start taking these acetyl-CoAs and kind of throwing them away since they're useless to you at this point, right? So that's both the end of beta oxidation, what we actually end up doing with that acetyl-CoA, and then what happens if we don't modify and use those acetyl-CoA, and what if we have to come back to it, right? So that's lipids. Lipids are easy. Lipids aren't too terribly complicated. You take that glycerol molecule, you'll deal with that glycerol molecule and just feed it into basically what is the last half of glycolysis. Proteins get a little bit more complicated. Well, what happens if you are starting to break down proteins? Well, step one is you generally don't. The solution to break down proteins is to not do it in the first place. But if you are somebody that is starving for any reason, and you are starting to break down stuff like skeletal muscle or liver protein, or God forbid heart protein, etc., that is starting to be a big deal, and that needs actually addressed pretty quick, right? The normal way that we break these down is we're going to intake a protein, right? I eat a steak or something. I intake that protein, which is a long chain of amino acids, right? We have lots of amino acids in a very particular order. That's what a protein is. I'm going to pass that protein, and it's going to go through my stomach. It's going to go through my duodenum. It's going to go through my jejunum, and it's going to be broken down into individual amino acids, right? We take those proteins and we're breaking them down to individual amino acids rather than having them be a long protein chain. Well, those amino acids are going to first and foremost be pulled out of circulation and taken up back through the hepatic portal vein. So this is where our hepatic portal vein comes into play, right? We have a shunt that puts all of this stuff directly into the liver, right? This hepatic portal system, this hepatic portal vein. What is the liver going to do with it there? Well, the liver is our site of major metabolic change. So this is where we undergo a lot of metabolic management of these sorts of protein molecules. So kind of step one is the liver kind of doles these out to whatever needs these amino acids the most. And because the liver is in charge of it, the liver gets first pick. The liver gets to take all of these proteins that it wants, which is actually generally okay, because the liver will use these to do a lot of important functions. The liver is going to do a lot of protein generation. We talked about things like albumin, right? Albumin, if you remember from way, way back, is a carrier protein. It helps with things like managing blood pH. It helps with things like holding liquid in circulation, holding like the plasma actually in the bloodstream. If you lose these liver proteins, then you lose these blood proteins, then you lose a lot of this onconic buffering effect where you have this sponge-like effect that brings fluid and holds it in circulation. This is also going to be things like haptoglobin, right? You've heard of haptoglobin. This is one of the things that's helping with iron metabolism. That's coming from the liver. Things like clotting factors are, not all of them, but most of them, clotting factors, as well as complement factors. Not all of them, but most of them. All of these complement factors are also coming from the liver, right? So the liver needs a lot of amino acids to build all of these new proteins that they're constantly using and breaking down. What else is the liver doing, right? The liver is also sending some of this off to other tissues. And when we say other, we really mean tissues mostly in the skin, right? Because I'm shedding skin all the time, right? I'm shedding not only my external skin, but I'm shedding the endothelial cells that are lining my gut, right? As the food is sloshing back down through, I'm shedding skin on the inside and passing that in my feces. I have to replace those skin cells, so I need a lot of protein to do that. I have to build a lot of protein cells. The other thing where we end up having a lot of this amino acids end up is the other major muscles are the other major amino acid repository in our body. Right? We have a lot of muscle, especially if you go to the gym and work out, you're building new muscle. What do you need to do that? You need amino acids to take that in the other direction and make those proteins. If you're somebody that is, your liver has quite enough, you're doing great, and you don't really have all that much muscle need, well, if you're in this case, and the last kind of case is, well, let's just break these down. Let's just digest them, right? So we're going to actually end up digesting these amino acids, and we're going to convert them into our energy sources. Some of these are going to go off and actually get reconverted back into a molecule of glucose. Some of these are going to get reconverted and get turned into a triglyceride. The big difference between them is what kind of amino acid that we're talking about. What does that amine group look like? Depending on what that amine group is, it will either become a molecule of glucose or a triglyceride, right? This is called glutagenic and ketogenic amino acids. I don't particularly care that you're familiar with which ones are which, but just the fact that I can make glucose by breaking down molecules of amino acid and digesting them, or I can make molecules of triglyceride and store them off later as fats, that's a nice thing to know and to be familiar with, right? So that's how we break these proteins down and what we actually end up using them for, right? If I want to break these down further and I want to actually digest them, I'm going to undergo, I have to change these molecules of amino acid a little bit, right? So we need to talk about what an amino acid actually is. An amino acid is, again, we're going to go back into our chemistry, but not too deeply. It's 2 carbons and then a familiar arrangement where we have some oxygens held on the outside, right? One of those oxygens is having a charge, right? Which is why this is an acid, right? This doesn't look too dissimilar to something like an acetyl group, right? And then we have on the other carbon, our old friend, one of these is there's another hydrogen, but then we actually have a molecule of nitrogen, and then that nitrogen is actually bonded to a couple hydrogens that are kind of hanging on. So we actually have an amine group. An amine that's hanging on this is why we end up calling these amino acids, right? This amine group, this nitrogen group, is what gives it its name and its designation. The last thing that's hanging off of this carbon, and I'll just draw the other hydrogen just for so that everybody's not freaking out about the number of carbon bonds, is what's called the R group, or the functional group, right? So this is the functional group of that molecule. What that ends up being, if I break this down, whether or not it's a ketogenic amino acid or a glutagenic amino acid, do I turn it into a lipid? Do I turn it into a glucose? Right, that is it's entirely driven by what this functional group is. If I want to break these down, one of the first things that I need to do is I need to get rid of this amino acid trash. So just to show this, right, Right, these are all of these functional groups. So these are going to tell you what these amino acids are going to, whether they turn into a glucose or whether they turn it into a lipid, is mostly being driven by what is the shape of this tail that's coming off of this functional group. Right, so what I want to do is I want to get rid of this amino acid. So I actually have to move this amino acid onto something else. So I transport, trans as in convert and change something, and I undergo a process called trans, as in like changing and moving, amination. Right? I transaminate this. What do I end up getting left with after I remove that amino acid group? Well, I'm going to take a 2-carbon chain with some oxygens attached to it, and then I'm going to have a functional group, right? So I've taken that molecule, and it now looks like kind of a run-of-the-mill 2-carbon chain with a couple oxygens linked onto it. This is the thing that will then, depending on whether it goes off and becomes a molecule of glucose or it becomes a lipid like a triglyceride, right? It's mostly driven by that functional group that's making that decision. So this is the thing I actually use for energy. These carbons are the bonds that I care about. But the transamination process isn't really over. I have to deal with that ammonia in some way, right? This amine, right? I need to actually deal with this. So I actually am going to cleave this off and basically create a charged molecule. But in doing so, I have to actually modify this and convert it into ammonia in a way that I can handle it. It can stay in circulation. It can be water soluble. So I actually do a couple little modifications to this, and the thing that I end up with is going to be a molecule of carbon that's holding on to a couple of these nitrogen molecules for me, right? So this carbon molecule is holding on to some nitrogen. Nitrogen is this nitrogenous waste, right? The stuff that I don't really want. I can't really deal with it. It's just trash to me. It has no function. I can't break it down and get energy out of it, right? So what do we end up doing with this? We take it and produce this molecule, which is a molecule that you might be familiar with, called urea. This is uric acid, right? This is our urea waste product. This is how I pee and excrete nitrogenous wastes, is coming out of this urea process. I have to transaminate that amino acid, and then if I am excreting lots and lots and lots of amino acid, if I have a lot of urea in my waste, well, that tells me that I'm intaking more amino acids than I actually need, right? I'm obviously not using these amino acids for anything if I'm transaminating them and breaking them down into fats or glucoses. So if I am excreting lots and lots of nitrogen, I'm going to be at a nitrogen surplus. I have way too much nitrogen. I'm going to make a bunch of urea. That's going to be the case for most adult healthy individuals that are eating lots and lots of relatively good for them food, right? You're going to have lots of amino acids. So if you're at a surplus of this, right, you are generating an enormous amount, depending on the degree of surplus, you're generating lots and lots of nitrogen. You're at nitrogen surplus, right? Most adults in normal healthy conditions are somewhere on this surplus, right? You don't need all the nitrogen, all the amino acids that you're taking in. If you are somebody that is growing quite rapidly, right? So you are taking in lots and lots of amino acids, but you're not actually transaminating all of them because you're using those amino acids right away to build proteins, to build skeletal muscle, to build skin, to build the cells that make up our rapid development and growth. If you're at this deficit, right, you're not going to be producing a lot of urea. There's very little urea that's coming out of the system. That's mostly for people that are undergoing a large amount of growth, right? If you're at a urea deficit, right? Mostly kids, people, if you're like working out a whole bunch, if you have like a rapidly growing tumor or something, right? That can be a trigger for a problem if you're at this urea deficit, right? You're using these amino acids as building blocks as opposed to breaking them down through this transamination process. That's how we actually can the nitrogen budget that someone is going through. We can use that as an indicator for health, right? So a picture, oops, jumped out of the slides, right? And everybody's going to get access to this, everybody's going to get points for these, right? So if I want to increase blood glucose, I'm trying to increase the amount of glucose that I have. I obviously don't want to undergo glycolysis, right? That's me breaking it down. I obviously don't want to create new molecules of glycogen, right? So I don't want to make new glycogen because I need to take glucose and produce glycogen from that. So I don't want to do glycogenesis. The C and D here are going to be cutting that molecule of glycogen, kind of that tree of glucose that we call a glycogen molecule. I want to trim that bush a little bit so that I have some of those glucose molecules falling off. Or if I'm undergoing things like gluconeogenesis, taking molecules like pyruvate and turning it into a molecule of glucose, taking a molecule of an amino acid and turning it into a molecule of glucose, right? That's undergoing gluconeogenesis. So both C and D would be the things that I would want to look at here. And just when we're talking about beta oxidation, the thing that we are producing from this is we are producing acetyl-CoA, right? We do make a lot of adenosine triphosphate, or ATP. We make a lot of ATP, but we're making that mostly is coming from oxidative phosphorylation, right? We're making those weird batteries, the NADHs and FADHs, pumping those into the mitochondria, using that to create a proton gradient inside the mitochondrial intermembrane space, and then getting ATP synthase to make a bunch. If I'm beta oxidizing, I'm not really making any oxaloacetate, right? I'm using oxaloacetate and pumping the Krebs cycle around, and I'm not generally producing a lot of glyceraldehyde 3-phosphate. That's the process where we take the molecule of glycerol, the other half of that triglyceride, and then turn it into another 3-carbon chain with a phosphate attached to it. So acetyl-CoA is the thing that we're making a bunch of through beta oxidation. So that was transamination. We added that. Now we can stop zooming in so much on chemistry, and we can zoom back out a little bit into just general metabolism and how we manage these, especially in times of boom and bust. Right, so if I am doing quite well, I'm going to store a lot of those glucose as glycogen. I can store those triglycerides in my adipose tissue, in my liver, right? I can start getting a nice fatty liver if I am intaking normally a lot of food. Amino acids are the weird one in that I don't really have the ability to store amino acids. This is why amino acid synthesis and protein synthesis is so finicky, right? If you don't have any reserves of it, you just can't make that protein. That becomes a problem, right? This is a very apt thing if anybody is like eating these amino acid rich protein things like whey protein, that kind of stuff. Anything that looks like somebody has smashed something through the label, it generally is these kinds of whey proteins. It's kind of a logo that they generally go with, right? Generally always looks like something's on fire or something is stamped into it to make it look tough, right? You are generally just urinating this stuff out, right? If you're not using these right away and you're not at a caloric deficit that you're going to need to make a lot of energy from them and actually go through transamination, you're mostly just peeing these amino acids away. Most of them actually end up being excreted this way. When you are looking at this, I want you to pause and go back to your notes for this. So like, don't immediately jump into this. But when you are taking these molecules, what this is trying to get at is which of these is the most energy dense. And if something is energy dense, it will generally contain the other things inside of it, right? So if I break down a bunch of fatty acids, that fatty acid will become a bunch of molecules of acetyl coenzyme A, right? So I already know that A is not the most accurate answer, right? A is not going to be a good answer, because if I have fatty acid versus if I have an acetyl-CoA, a fatty acid is lots of acetyl-CoA. Other thing, a triglyceride has 3 of these, right? So if I'm just looking at a generic fatty acid and a generic triglyceride, I have at least as much energy inside a triglyceride as I do inside of just a single fatty acid. Ketones are a little bit different, right? Ketones don't really fit on this chart, because if you have a ketone is this kind of, I taken a molecule of acetyl-CoA and I've split off the CoA off of it, and the thing that I'm left with is I'm left with that keto, and left with that ketone or that keto acid. I can reconvert this keto acid back into an acetyl-CoA if I put a little bit of energy into doing that, but that's obviously pretty wasteful, and it's no better or worse than just a regular acetyl-CoA, right? So you can kind of think about keto acids as being like a worse version of acetyl-CoA. We have some limited capacity as humans to convert between molecules of amino acids. So if I'm intaking something with a lot of methionine, but I'm not intaking something with a lot of cysteine, the fact that I'm probably intaking enough homocysteine in my diet means that I can pretty much, as long as I'm relatively healthy other than the fact that I am cysteine deficient, I'll probably be able to produce some of that from homocysteine, right? You need some help along the way. You need some vitamins to help and act as coenzymes to help this process go. But some of these amino acids are not as difficult for us to go through. If we have a relatively well-rounded diet, you're not going to be amino acid deficient for any one thing. These sorts of amino acid deficiencies are only going to show up when we're concerned with people that are on very limited food choices, as in like you only eat rice, right? You only eat cassava. You only eat white bread, right? Not even like nutritionally dense bread, right? This helps us and keeps us from being suffered by most of the generally bad effects of not having the correct amino acids. I'm not going to draw homocysteinuria because we don't. That's a little bit more than I want to get into the weeds right now, right? So we're not going to worry about too much about that. Let's talk about fed and fasting, though. When we are in a fed state or a fasting state, you should view this as a broad hormonal regime. Insulin and glucagon is the simple way to describe these, right? Whether if you have a bunch of high blood sugar, you're in a fed state, so you have a lot of glucose reserves readily available. The dominant hormone is going to be insulin. You're in the opposite end of things. It's been a while since you've eaten. The glucagon is going to be the thing. Your glucagon, you're trying to up your blood glucose concentrations, right? If you're fed, you'll last for a couple hours. And the way that this is going to work is you're going to be pulling all of that energy out and then storing it elsewhere, right? You're storing it mostly as glycogen in places like the liver, as well as in skeletal muscle. And then you're storing it in temporary reserves in places like triglycerides, right? You're taking those triglycerides in both the liver as well as adipose tissue and storing that and holding on to it for later. In the fed state, we're also going to make a little bit more protein, right? Like you can see right here, your skeletal muscles are going to grow faster if you've just recently had a meal. Your liver is going to amp up clotting factor production if you've just recently had a meal. But this is not going to be a nice energy source because I really don't want to break those proteins down later on. That means that you've kind of goofed up and you've gone and starved yourself for some reason. So if you're fed, pretty much things are flowing from the alimentary canal. Energy is flowing out and then being either immediately used for cellular growth and cellular division, specialization, or we're going to store this off for later in the form of like glycogen and adipose tissue, right? Insulin's driving this for the most part, right? You've seen this diagram before. I'm starting to add a little bit more complications into this because now you actually know what pyruvate is, right? Insulin tells these cells to say, hey, there's way too much glucose outside. You're hyperglycemic, right? There's too much glucose in your blood. Let's suck some of that in and then either ramp up glycogen production and store it in the form of glycogen, or we can actually take these pyruvate molecules and produce fatty acids from them. So both of these are things that are ramping up in activity because of the actions of insulin, right? So you're going to have more adipose tissue if you have a high insulin concentration. You're going to have more glycogen being produced if you have high insulins. That's not only what it's doing, right? This is also doing things like saying, hey, this is a boom time. We have energy, so let's do all the stuff that we want to do with that energy. Let's start ramping up protein generation. Let's say, hey, I don't want you to make new glucose, right? I want to inhibit gluconeogenesis. I don't need any more glucose. I've already got plenty, right? So it's doing things like stimulating more protein generation, more amino acid use, as well as stimulating the production of glycogen, but it's also ramping down the activity of some of these actions, right? I don't want my liver to start upping my glucose concentrations if I have insulin, because insulin's only going to be released if I already have high glucose, right? That's when things are going well. Much more commonly, in historic times, we would go through a period of fed and fasting. We don't really have this problem, most of us in the developed world, right? So we're snacking perpetually throughout the whole day. So we're generally in the fed state for most of the day. But if we go a while, if we go to sleep, or we actually refrain from eating between meals, that kind of thing, then we're going to tip into starting to go down into our glucose reserves, starting that the glucose that's just circulating in blood starts to be tapped out. I'm using that glucose all the time to make sure all of my cells are running. So whatever glucose that I have in my blood, I have to replenish. I have to use that glycogen stores, release that glycogen, or I can switch to something that's not glucose if I have to. All right, so let's talk about how we're doing this in the fasting state. I think this is going to be our last little diagram. Yeah, I think so. Right? If we are in the fasting state, there's various degrees of fasting that are going to come along with how severe the fasting event is, right? But if we're in a fasting state, this is going to include anything from a couple hours on to a couple days or weeks or months on, right? Fasting is a pretty big range, right? So if you're fasting, what's happening is you're starting to become hypoglycemic. We're starting to become hypo as in not enough, gly as in glucose, as in like sugars, semic as in glycemia is in the blood, right? So we start having low blood sugar. That low blood sugar is a pretty profound stimulus to tell our pancreas, right? Our pancreas is going to listen to this, and our pancreas is going to respond to this hypoglycemia saying, oh no, something's really wrong. I need to fix this. I'm going to start pumping out glucagon. Glucagon, being the dominant hormone here, means that as glucagon starts to rise, if you kind of chart this over time, if you're like looking at how much glucagon, right, we're going to have big normal variations, but if I start kind of starving myself, my glucagon concentrations are just going to start going up and up and up and up and up, and will continue to rise until I deal with the blood sugar problem. If I'm always hypoglycemic, I'm going to continue to stimulate and produce more glucagon. Well, as glucagon starts to rise, right, as glucagon levels start to go and tip above where they normally are, what's the first thing that I'm going to do? Well, the first thing that I'm going to do is I'm going to start ramping up and stimulating glycogenolysis. Right? I have these long chains of glycogen, and I want to start trimming those glycogen trees. I'm going to trim those glycogen bushes and turn them back into glucose. All right, where is this taking place? Again, this is mostly taking place in your liver, and this is mostly taking place in your places like your skeletal muscle. These are going to be the dominant places where you have a lot of glycogen stores. This is going to work for a couple let's see, maybe hours to kind of the end of the first day, right? You're going to be responsible for most of that glucose concentrations going up is because of those glycogen stores. As glycogen stores start to be depleted and you start to not being able to regulate blood sugar based purely off of this glycogen, you are going to continue to see a rise in blood glucose. Blood glucagon, right? Glucagon is going to continue to go up and up and up. Well, what do you do as you start hitting these kind of increasingly concerning periods? You're going to undergo the second major transition. You're going to start ramping up and stimulating the additional lipid breakdown. This is where the lipids start to be used. You undergo lipolysis. We take those molecules of triglyceride and we cleave that molecule of triglyceride and turn it into fatty acids or free fatty acids and a molecule of glycerol. Right? As those glycerol molecules get broken down, we turn them into glyceraldehyde 3-phosphate. We run that glyceraldehyde 3-phosphate into pyruvate. We turn it into a molecule of acetyl-CoA, and then run it around the Krebs cycle. What else do we do with this fatty acid? Well, we just skip a couple steps and turn it directly into acetyl-CoA, and then run it around the Krebs cycle again. Pretty energy dense material. It's very nice to use. It's easy for us to have a lot of energy stored in a relatively small amount of tissue if we're undergoing lipolysis. This is what we're going to do on the order of kind of a 18 plus hours, right? If it's been a while since you ate, or you're starting to ramp up cellular activity, like you're working out for a long time at relatively slow rates, you can still do a lot of fat burning, because this is going to be on the order of kind of a few hours to a couple days, a few days. Generally, we're talking about a little under a week, right? Most of us have enough lipid stores that we can increase our blood sugars with a little bit of lipolysis. It's okay to do that. Notably, the only way that I can actually increase my blood sugar is I can actually take this molecule pyruvate and turn it back into glucose. I actually can't take a molecule of acetyl-CoA and turn it back into a molecule of glucose. I don't have the enzymes to make that process go. So this is why relying on just the acetyl-CoA is actually still an okay thing. We're going to talk about the brain, why we want to be especially careful about leaving some glucose for the brain, because the brain's a very picky eater. Well, okay, you've undergone lipolysis, you're starting to mobilize fat reserves, you're still starving, you still are not consuming any additional food. Also, notably, this is taking place mostly in the liver, as well as adipose cells. Again, the liver is going to show up in a lot of places. So things continue to get worse and worse and worse. Glucagon levels continue to rise. They never keep getting pushed down and staying down because we are always hypoglycemic. That will always be causing the pancreas to make glucagon. So the next thing that we're going to start to go to is, as we start to go further and further into starvation, is the bad one. The one we really would like to avoid. We would like to avoid cell protein catabolism. I don't have just a bunch of amino acids laying around, right? I don't have a bunch of protein laying around that doesn't already have a job. That protein is doing something. So when I start to catabolize, as in like break down and break those proteins and get the amino acids out of them and then collect the energy from those, this is a big problem. This is kind of, we're looking at like weeks of non-eating or weeks of caloric deficits are going to start leading to things like cell protein catabolism, right? We're going to do a lot of things like transamination, right? We're going to transaminate, right? Where we're going to break down that muscle that's contained within our bodies. We're going to break down those proteins in all of our cells. Every one of our cells is going to run on this. So we're going to transaminate and then eventually get some things like triglycerides or glucoses out of this. So this is nice. It's a way to up my blood glucose, but it's a pretty big cost that I'm paying. Because mostly the places where you're going to eventually see this happen is the first major site for the way that the first major place that we start doing a lot of this catabolism is going to be skeletal muscle. You're going to start seeing a person becoming very, very thin. Their big muscles are going to go away. The next place that you're going to start to see as the major site is once you've started to dip into your skeletal reserves and that starvation period continues, you start to hit smooth muscle, right? So that smooth muscle lining places like your alimentary canal itself, smooth muscle wrapping around all of my vasculature in my blood vessels, around my heart, right? As well as the big one, cardiac muscle. Why do we want to have the cardiac muscle be the end point, right? I don't want to dip into cardiac muscle as a big energy source first, because this is how you die. This is how you die from starvation. You don't die from just not having food. You die from heart failure. You die from heart failure because you are actually consuming so much of your heart's cardiac muscle that your heart can't pump strongly enough anymore, and this ends up actually causing death. The smooth muscle breakdown is a big problem as well. This is the reason why refeeding syndrome exists. If you are looking at patients that are starved for long periods of time and then you give them a large meal, you can actually kill them because you've given them a meal that their intestines can't handle. They don't have very strong actions of peristalsis or segmentation because you don't have much smooth muscle left. Right, that's why places like homeless camps, people with mental illness that are going long periods of time without eating, you see a lot of this happened in various concentration camps in World War II or concentration camps since, where people are all of a sudden get a little bit of food again, and then all of a sudden they die. Part of the fact is because of the smooth muscle loss. They've eaten away their ability to have a functional digestive system. Right, so this is why we have cardiac muscle and smooth muscle are really at the bottom of the bottom of the bottom of the list. Glycogen is very easy to break down. I have it specifically for this function. Lipids are pretty easy to break down, but again, I have lipids on hand as triglycerides, mostly for the rainy day situation. Cell protein catabolism is you breaking functional tissue in your body down. I am losing function as I'm trying to survive another day, right? So that's what we're trying to avoid. The reason why we have to be careful and the reason why glucose isn't always entirely sucked up by tissues even when glucose is available is we do this thing called glucose sparing. And this is weird. This is not something that is well understood why this happens, or we understand why it happens, but we don't understand why the brain's so darn bad at this. Our brain cannot use a triglyceride. It cannot break down triglycerides and use them as an energy source well at all. It's very, very inefficient. It's very, very slow to do so. So we pretty much are relying entirely off of glucose. If we become hypoglycemic, as your blood glucose concentrations go down, all of your other body systems are going to start switching to things like lipolysis and, if they have to, blood protein or protein catabolism. But your brain is pretty much stuck at that state. Your brain has to have glucose. This, combined with the fact that your brain is very hungry as an organ, right? So this is a relative amounts on this chart. This is 10 times the amount that you would expect the brain to actually be consuming. Our brain, we have paid very much for this fact that we have very large functional brains that help us do things like go to the moon and take anatomy and physiology courses, but there's a cost to it. We actually have to pay a lot of energetic resources to this every single day, right? Why do we have this system where the brain can't do it? We know the brain can't. If we could understand why this is happening, why we have these other systems, it's probably a lot of these all together. Notably, I can't really immediately turn on a whole bunch of blood glucose availability when I am running away from a bear, right? I have to kind of mobilize other resources like glycogen. It's very easy to cleave glycogen off. It's very fast to do so. But even then, it's really hard to get things past the blood-brain barrier, the BBB, right? The blood-brain barrier is this setup that we have in our brains that is very nice and protective and keeps things like pathogens out of our brain. Very good. Not a fan of pathogens in the brain, but it also makes it a one more step that we have to get over in order to push resources like nutrients into the brain itself. And then we're going to talk about cholesterol and cholesterol metabolism another time. So this is going to be all we go to today. We're going to do cholesterol. We're going to finish digestion on Friday, and then we're going to jump into kidneys and finish kidneys. We're so close to the end, everybody. All right, I'll see everybody on Friday.